All right, this is the second lecture for the genes and genetics section of this course. And what we're going to do today is we're going to look at a few definitions first before going into talking briefly about different types of disorders, what these two terms mean, and as well as talking briefly about multifactorial conditions. So just to start off with, we know that the normal human cell has 23 pairs of chromosomes, and out of the 23 pairs, what happens is you have 22 autosomes, and autosomes are basically not sex linked, and then you have one pair of sex chromosomes. And normally sex chromosomes, either you have two X's or you have an X and a Y. And the two X's are found in females, and then males have an X and a Y. And if you look over here in the sex chromosome, so this is an X as well as a Y, you look at how much material the Y has compared to an X. And so we're going to talk about sex-linked conditions later on. And over here you can see that if a male with one X and one Y chromosome, the material that actually is found in the X is quite a lot relative to the small amount of material in the Y. And so that's why males have got more sex-linked conditions, because the Y is not protective for whatever mutations or genetic conditions that are found in an altered X condition. All right, so the picture in the middle is an example of the seventh chromosome. So you've got seventh pair of chromosomes. So you've got chromosome 1 and 2, and over here what happens is each of these different specific locations in the chromosome is called a locus, right? It's called a locus. So each locus would is responsible for different characteristics that make up a person. And so what happens is if you have variations of, say, this locus over here, they would mean that these would be heterozygous. If, say, this locus over here, let's say it's BRX locus, and say that the one on the left is BRX positive and the one on the left might be BRX negative, so they're different, and so they would be heterozygous, right? If the genes at the locus is the same, they would be homozygous, and so you would have two either BRX positives or two BRX negatives. Right? I'm just taking it out of a hat. And so these different variations, the BRX positive and BRX negative, are called alleles or alleles, depending on whether you speak American or British. And what happens is that it's the combination of maybe these two alleles at the same locus that might actually give rise to a genotype and phenotype. So genotype is basically what genes you have. So for example, if a person, when we look at the blood type, a person with either two A's or a person with two O's might end up with the same phenotype. And the phenotype of a person with either two A's or an A and an O is that the person would have type A blood, right? So this would be the phenotype, and this, the two A's and the A and an O, would be the genotype, right? Another example is if a person has brown eyes would be the phenotype. And a person with brown eyes can inherit one brown-eyed gene from one parent and one blue-eyed gene from the second parent, or can have two brown-eyed genes, right? So this genes that they inherit from the parents is the genotype, whereas what they show, which is brown eyes, is the phenotype, right? And so you've got over here, this is a chromosome 9. You've got just different locus on the chromosome 9, so the ABL and the Q34. And over here, I've got just the definition of the terminology that I've spoken about, explained here in a little bit more detail. Next slide, you've got, again, some terms. So here we're going to talk about dominant and recessive genes. So you've got, so genes, as I said before, come in pairs, right? So what happens is that we inherit one gene, one chromosome of each pair from either parent, right? And so what happens is you can have genes that are dominant and genes that are recessive. So we're going to look at later on about dominant disorders versus recessive disorders. And so a dominant gene would be expressed. So if you have a dominant, say a dominant D gene, that trait that's found within the dominant gene would end up being the phenotype, right? And it doesn't matter, say this is the recessive trait. If a person has capital D, small d, the phenotype would be the big D. Whereas if you have a recessive gene, what happens is that in order for the phenotype to be the little d, that person would have to inherit a little d from each parent. And again, we're going to go through it as we continue on in the lecture. So someone with a dominant disorder only has to inherit it from one parent, right? And so an autosomal dominant disorder would be non-sex based, and so would affect both sexes equally. And we're going to talk about X-linked dominant disorders later on in this lecture. A recessive disorder, as I said, needs to be inherited from both parents. So an example of this is sickle cell anemia. So sickle cell anemia, a person with sickle cell anemia would have inherited the sickle cell gene from both parents. If a person only inherits the sickle cell, the small s gene, from one parent, they would be a carrier, but they would present themselves as having normal hemoglobin. All right, so recessive disorders need to be inherited from both parents. Dominant only need to be inherited from one parent. Recurrence risk is basically what are the chances of a child having a certain disorder. Now, the chances of the child having a certain disorder is the same regardless of how many children a couple has, right? So for example, if you have 8 children and 4 of them have got this a disorder which say affects 50% of or has a 50% recurrence risk. So if you have mom and dad and they have 8 children, and they these 8 children have a 50% recurrence risk of inheriting disorder X. What happens when they have the 9th child is that the 9th child will also have a 50% chance of inheriting the disorder, right? So regardless of how many children, say 3 out of 8 children, if 3 out of 8 children have the disorder, it doesn't mean that the 9th one is definitely going to have the disorder. If 7 out of 8 have the disorder, doesn't mean that the 9th child will have no chance of having the disorder. So the recurrence risk for each child is identical regardless of how many children the couple already has, right? So with autosomal dominant disorder, if you have one parent that has the disorder and one parent that does not, what happens is that the recurrence risk is 50% for each child, regardless of how many children the parent already has, right? And that's because each birth is an independent event. It's not, so each child that is born has an identical recurrence risk, right? So for each of the 8 children in the 9th, they would have a 50% chance of getting the disorder if the recurrence risk is 50%. All right, let's go through this. So what we're going to do, and again, this is just background information. So these are pedigrees. So these are just common family tree diagrams that are used. And so you see over here, if there's a circle, it indicates that it's a normal female, a square, normal male, a diamond, the sex is unknown, a single bar connecting 2 people, a male and a normal female in this case, indicates that that they've mated. And over here, further down, you've got a normal male, normal female mating, and they produce 3 children, right? So this is first generation, this is second generation, and you've got 1, 2, 3 kids. And then, so if you have a single parent, doesn't mean that these 2 girls are formed from 1 male. It just means that the mom is normal and therefore has no significant impact on the outcome of the children, right? Double bar means that it's mating with a close relative, so like a cousin, first cousin, second cousin, third cousin, or between siblings or what have you. And this means that it's fraternal twins, so siblings, non-identical siblings born at the same time, whereas this indicates that that it's an identical twin. All right, so again, we're going to use some of these terms as we go along. All right, so what we're going to do is we are going to talk about, start talking about different types of disorders. So the first type of disorder are single gene disorders. So these are conditions that are indicated by just one single gene, right? And over here, you've got a dominant disease. So it only takes one from one affected parent. And autosomal means it's not sex-linked, right? And so what you have over here is you have the dominant gene is the D, disease gene, normal gene is the little d. And so the genotype of a person with the autosomal dominant disease would be big D, little d, or big D, big D, right? Genotype of a person without the condition is when they have 2 normal genes. Okay, so what you have is you have an instant on the left when you have both parents are affected. And note how I say parent, right? Because it doesn't matter whether this is the mom or the dad, or whether this is the mom or the dad, because these are found on non-sex linked genes. Okay, so this is the disease gene, and the little d is a normal gene. All right, so let's go to it. So say they have 4 kids. What happens is that you just combine. So from, say, parent 1, they inherit the little d. From parent 2, they inherit the little d. So you have 2 little d's, right? So out of this, what happens is out of 4, you have 3 children that are affected. Oops. So you have 3 children that are affected, right? So what happens is that the recurrence risk is 75%, right? Risk of being carrier, there are no carriers in autosomal dominant disease. Risk of being normal is only 25%, right? Males versus females, same risk, okay? So another condition, another instance is when you have a normal parent with 2 little d's and you have an affected parent with 1 affected gene and 1 little gene. So again, if child number 1 inherits big D from parent 1, inherits little d from parent 2, Child number 2, again the same, big D from parent 1, little d from parent 2. Child number 3, little d from each parent, and child number 4, little d from each parent. So over here, what you can say here is out of the 4 potential children, right, you have 2 children that are affected, so 2 out of 4. So the recurrence risk is 50%. Okay, and again, risk of being carrier, there's no such thing as a carrier for a dominant disease. Risk of being normal is 50%, and males versus females carry the same risk. Okay, so hopefully you'll understand this. So this is an example. This is what happens when you have an autosomal dominant disease and it's passed down. So some examples of autosomal dominant disease is Huntington's disease, right? So you have to inherit it from one parent, and that will lead to signs and symptoms related with Huntington's disease. So an example, so just some rules of autosomal dominance. We have here males and females equally affected. And if they're affected, they're likely to transmit it to half of their offspring, both males and females. Luckily for us, autosomal dominant diseases are rare, right? Most common occur in 1 in 500 individuals. So it's uncommon for 2 individuals produced by the same autosomal dominant disease to produce offspring together. So what it's saying is that it's very unlikely if you have a specific autosomal disease that you will actually mate and have children with someone with the same autosomal dominant disease. Okay, and there's no skipping of generations, and we'll look at skipping of generations later on. So it's hardwired. So if you have an affected person, like you see over here with the red circled line, so this is generation 1, 2, 3, 4, then what happens is some of the children may have it, right? So it does not skip a generation. So let's look at the family tree for achondroplasia. So achondroplasia is when they have a malformation of the cartilage, and so you end up with and it's an autosomal dominant disease. So this little girl has got achondroplasia. So again, shortened limbs relative to trunk length, the length of their torso, their body, and you've got other general phenotypes associated with achondroplasia. All right, so mom has it, right? Circle is female, colored in means it's affected. And they have 1, 2, 3, 4, 5, 6, 7 kids. And out of the 7 kids, 3 out of 7 have it, right? Their children then have grandchildren for them. And again, we're just taking children for child number 5, right? Because child number 5 is affected. And you can see here, child number 5 has 1, 2, 3, 4, 5, 6, 7 children, very convenient. And out of the 7 children, 3 have it. And you can see over here that in each generation, both males, squares, and female circles are affected, right? And again, when we look at the 4th generation, the great-grandchildren, what happens is you can see here out of the great-grandchildren, if the person has it, right? In this case, the dad mates with a normal female, and out of the children that of this 3rd generation, you have 1 out of 3 affected. The 2nd child is normal, mates with another normal individual, so you have no children affected. 3rd child is affected, right? Mates with a normal individual, 1 out of 3 affected. 4th child is affected, produces 2 offspring, 1 out of 2 is affected. 5th child does not produce any offspring. 7th child also doesn't produce any offspring. Child number 6 mates, has 2 children. Child number 6 is normal and produces no affected children. So what you can see here is that there's a chance, if you're affected, that you produce affected children, 50%, right? And if your child inherits the normal version of the gene, then there's no chance at all that they will actually pass it on to their children. All right, so if neither parent has the trait, none of the children has it. And then you can see this between the third and the fourth generation. Next disorder is autosomal recessive. So autosomal, again, non-sex linked. Recessive means you have to inherit 2 copies of the gene for the disorder to manifest itself. And we talked about a disorder that is recessive. Sickle cell anemia has to be inherited from both parents, right? So in this case, what happens is the recessive gene is the disease gene, right? And then the normal gene is the capital D. Okay, so again, so what you have is you have a normal parent and you have a carrier parent. So a carrier parent phenotype is normal, right? A carrier parent does not have sickle cell anemia because a carrier parent would have 1 affected gene and 1 normal gene, which means that they don't have it. Okay, so genotype of a person with a condition has to be 2 small d's. Genotype of a person without a condition can be either 2 d's, 1 d, 1 big D and 1 little d. Okay, so let's go to it. So you've got a normal parent and a carrier parent. So you do this. All right, so you have a normal parent, a carrier parent, recurrence risk 0, risk of being a carrier 2 out of 4, so 50%. Risk of being normal, phenotypically normal, would be 100%, right? Or if you have a normal genotype, would be 50%. Males and females, same. If you have a condition where you have 2 carrier parents, so again, this is the affected gene, and this is the normal gene. So let's do this. Okay, so again, when you look at this, genotype of a person with a condition is this. So 1 out of 4. Right? Recurrence risk is 25%. Risk of being normal, if you look over here, of being the carrier would be 2 out of 4. So that would be 50%. And I'm going to revise my definition of normal. So normal means phenotypically normal. And so risk of being phenotypically normal is 1 out of 4. So that's 25% as well, right? Males versus females, same. Over here, what we have is we have an affected parent and we have a carrier parent. And this is actually quite common because a carrier parent would appear to be normal, right? Would appear to be phenotypically normal, and so wouldn't know that they actually have that recessive gene in them. All right, so again, let's do this. So you've got 1 D, 1 big D, little d, and then you've got that. So again, if you look over here, if you look at whether or not they will have the condition, it's 2 out of 4. So 50% will have the condition with 1 affected parent and 1 carrier parent. Risk of being a carrier is this, so 2 out of 4. So that would be 50%. Risk of being normal, 0. Males versus females is the same, right? So don't have a chance of being normal. They either are a carrier or they actually have the condition, the autosomal recessive condition. So just some rules of autosomal recessive disorders. So again, rare in populations, although the numbers of carriers for recessive disorders can be high because the person doesn't know they have the disorders, right? Males and females are affected in equal proportion, so it doesn't matter whether you're male or female, you have the same recurrence risk of getting or not getting the disorder. Consanguinity. So if you have this double line over here, so this is a mating between two close relatives, between a female and a male, what happens is it increases the risk of a person getting it, right? That's there. Disease is usually seen in siblings but not their parents, because sometimes their parents can both be carriers and not have the disease. And so it would actually show itself in the parent, right? 25% of carrier parents will be affected. So again, if we go back, you've got here, right? If you have two carrier parents, then you have a 25% chance of getting the disease. And then sometimes it's characterized by delayed onset, incomplete penetrance, and variable expressivity. So we're going to look at expressivity and penetrance later on. Delayed age of onset means that for some of the disorders, the person doesn't get it as soon as they're born. So it might develop over time. So the person might live till they're in their 30s or 40s before the disorder actually manifests itself, right? So an example of an autosomal recessive disorder is cystic fibrosis. So cystic fibrosis commonly is an autosomal recessive disorder, and you can have two parents who are carriers for it, seem normal, don't know that they are carriers for cystic fibrosis, have a kid or two, and one or both kids may have the cystic fibrosis. Okay, so cystic fibrosis occurs in 1 in 2,500 births, 2,500 births. But in terms of the Caucasian population, 1 in 25 Caucasians, males and females, are phenotypically normal and are carriers. So they only have one copy of the affected gene. So you see over here, you've got homozygous, so same from both, and they're affected, and then you've got carrier parents, right? So this is parent 1, which is a carrier, and parent 2, who is also a carrier, and they have a 25% chance of actually producing an affected offspring. Next thing, sex-linked disorders. So again, if you look at the karyotype in the first slide, you have an X chromosome, which is quite large, and then a Y chromosome is quite relatively small. And so the Y chromosome only contains a few genes, and so most sex-linked traits are located on the X chromosome and therefore are said to be X-linked or sex-linked, right? If a male inherits a diseased X chromosome, what happens is that male will actually have the condition because the males then don't have another X to actually balance it out or to mask the abnormal gene, right? So X-linked disorders can be either dominant or recessive, and if you have an X-linked dominant disorder, an affected parent, an affected father who's XY will always pass the affected X gene to the daughter, because remember, daughters have two X's and males have a Y, and the Y gene for an affected parent is normal, and so the sons won't have the disorder from the dad. All right? An affected mother would pass the condition to 50% of her children, because both her girls and boys, both the daughters and sons, have a 50-50% chance of inheriting either one of the X chromosome from the mom, right? And half of the and one of two of the X chromosome is affected, the other one is normal. So 50% chance of her passing it to both her daughters and her son. X-linked disorders mean X-linked recessive disorders means that it needs to be found in either both X chromosomes for a female, or for a male you only have one, right? So these are people with a condition, with X-linked recessive conditions, and so you can imagine that mothers will pass their condition, affected fathers will again transmit the condition to all his daughters, because the single X gene that he has is affected, but not the son, because the son gets the normal Y gene. Affected mother would pass the condition to all her sons, because both are affected. And all her daughters will be carriers, assuming that the father is normal, right? And if the mother is a carrier, they would have one affected X chromosome and one normal one. And so they would pass their condition to 50% of their sons, and 50% of their daughters, 50% of the daughters will be carriers. Okay, so let's do this. So you've got X and Y genes, because it's sex linked. So the capital H is normal, the small h is a disease gene. So a carrier for there's only carriers in the for girls, right? A carrier would be someone with one, whereas an affected person for a girl would have this phenotype, and for a boy would have this phenotype. Okay, so let's do this. So you have a carrier mother and a normal father. And then you'd have this. Okay, so recurrence risk, if you look at how many people are affected, so you've got this, which doesn't can't come about with a carrier mother and a normal father, but you also have that, right? So out of the boys, you have 50% sons and 0% daughters, right? Risk of being a carrier only exists in the daughters. So 50% of the daughters are carriers. And of being normal, you've got 50% son and 50% daughter. All right, so males versus females, you can see here is very different. So more for sons, right? So 0% of the daughters is not likely to have the condition, whereas half the number of sons have the condition, or 50%. There's a 50% recurrence risk. All right, so if you have a normal mother and an affected father, you have this. and then Y and X, Y. So again, the sons of this couple, because the Y gene does not carry the affected chromosome, therefore the sons will all be normal, right? If you look at the affected father, if you look at the daughters, what happens is all of them will be carriers, right? So recurrence risk is 0 with an affected dad. Risk of being a carrier, 100% for daughters. And risk of being normal is 100% for the sons, right? So again, males versus females. The affected father won't pass it on to his kids, but then in several generations it might actually come about if the carrier mom actually mates with an affected father, as we can see in the next box. So you've got carrier mom, so one normal, one disease, affected father, single X disease, right? So again, let's do this. So again, risk of being a carrier, only have carriers for the females. So this is a carrier, right? Risk of being normal would be this one. And then risk of actually getting the condition is this and this. All right. So again, if you look at the overall recurrence risk, overall is 50% overall. Risk of being normal, risk of being a carrier is 50% for girls. Risk of being normal is 50% for sons. And you see over here, males versus females, you've still got 50% recurrence risk for both males and females, but in terms of whether or not the other 50% is carrier or normal, differs between males and females. X-linked dominant disorder, carrier, there's no such thing, because if you get it, then you have it. So X-linked, the disease gene is the capital H, and so an affected person would have either that phenotype or that phenotype for that genotype for a girl. Oh, sorry. Yep. Okay, so let's do this. So affected mother and normal dad. So you've got this. And then and so what happens is there's no carriers. So no carriers. I'm going to write it all no carriers, because you're either affected or you're not. And so risk of being, actually what I'm going to do is I'm going to do all of it. So you've got a second instance where you have a normal mother and you have an affected father. And then you've got affected mother and affected father. And so let's do this. All right, so risk of being, of having the condition, you have this, so that's that, and that's that. And then you also have that, which is that and that. All right, and then that as well. Okay, so recurrence risk, if you have an affected mother and normal father, recurrence risk is 50% for girls and 50% for boys. Normal mother affected father is 100% for girls, 0% for boys. And affected mother, affected father, 100% for girls and 50% for boys. Okay, and then I said there's no normal, and so the rest, so 50% chances of being normal is 50% girl, 50% boy. If you have an affected mother and a normal father, normal mother affected father, risk of being normal is 0 for girls and 100% for boys. Whereas if you have both parents affected, then risk of being normal for girls is 0 and 50% for boys. And so you can see that in these cases that there are differences between the males versus females in terms of the percentage recurrence risk of getting the disorder. A couple of other things. So penetrance. So penetrance is basically someone who has the phenotype that has the genotype, right? So someone, say for example, a condition can have either complete or incomplete penetrance. So a condition that has complete penetrance means that 100% of affected people, 100% of people with the affected genotype have the exhibit the phenotype, right? So all individuals with a gene express the disease. With incomplete penetrance means that only some people with the gene actually express the disorder, right? So for example, if you have the gene for, I don't know, condition X, and condition X has incomplete penetrance, that means some people actually end up with condition X and other people end up without condition X. They look normal. Whereas say if a person, if condition Y has complete penetrance, that means everyone with the gene for condition Y ends up with the disease that is expressed by condition Y, right? Some conditions are age-dependent, exhibit age-dependent penetrance. So for example, Huntington disease is an autosomal dominant disease, but people are not born with Huntington disease. Huntington disease usually strikes when a person is in their 30s or 40s, and so they can develop quite normally as a child and as a young adult, and then they get signs and symptoms of the disease when they're in their 30s or 40s, right? Breast cancer is another instance of age-dependent penetrance, and so is polycystic kidney disease. All right, so an example of a condition with incomplete penetrance is retinoblastoma. So retinoblastoma is an autosomal dominant disease. It causes an eye tumor in children. And what you see over here is the filled in red circle or squares are people with the with a genotype and with a phenotype, right? People who have the gene and who have the condition. And so what you see here is that retinoblastoma has an incomplete penetrance of 90%, right? So only 90% of people with the gene for retinoblastoma actually develop retinoblastoma. The other 10% seem normal, so they don't have the disease, and they are called obligate carriers. And so an example of the person with the genotype that does not have this disease is this female with this arrow on it, right? And we can say that this person does not have the phenotype, even though they have the genotype. And lo and behold, when they have a child, when they have a girl, the genotype is then passed down, and then her daughter would actually exhibit the condition, right? So this is an example of an incomplete penetrance of a condition. Expressivity is how that condition is expressed. So what if you have a gene for a certain disease, it does not necessarily mean that everyone with a gene for a certain disease has the exact same signs and symptoms and condition. So an example is neurofibromatosis. So neurofibromatosis, a person with the gene for neurofibromatosis can end up with basically dark spots on their skin, which doesn't affect function of that person, or can end up with this malignant neurofibroma, right? It can also affect their spine. They end up with scoliosis, can end up with tumors of the glial cells, or tumors of the neurons, can end up with hypertension, can end up with learning disorder. So the different ways that the condition is expressed is called expressivity, right? So how it works is that a parent who has these dark spots may not actually realize that they have the gene for neurofibromatosis. And so they may not be aware of it, and they can actually end up with a child that has this severe version of the condition, right? And so what happens is you've got the spectrum disorder from mild to almost non-existent to something that's actually quite severe, like high blood pressure, learning disorder, tumors of the glial cells and the neurons, and disorders of the spine, right? So the reasons for the variability might be that they have other genes that actually affect the condition and cause the modification of the expression of the condition, right? Or it might be environmentally mediated, or there might be a slight mutation versus a more severe mutation that causes the differences in the expressivity of the condition. Last thing we're going to talk about is multifactorial conditions versus polygenic traits versus conditions that have an environmental and lifestyle component to it, right? So polygenic traits are traits that arise when you have multiple genes acting together, all right? Multifactorial traits are variations of traits that are caused by genetic and environmental or lifestyle factors. So for example, the height of a person can be roughly determined by looking at the height of each of their parents, right? So you would say that on average, the daughter would be about the same height as the mom, and the son would be somewhere in terms of height would be somewhere as tall as somewhere in between the mom and the dad, say. But there are other factors that actually play a part into how tall a person ends up being. And part of it is nutrition, right? So the more you eat, the bigger you are, more or less. And also access to healthcare means that if you have a sickness, it can be easily, and it's quickly treated, then that sickness or illness will not affect the child's growth and development, right? Another example is IQ of a person. So an IQ of a person can be determined by a person's genetic origin, so the mom and the dad, but it also can be dependent on the environment. So either the child grows up with tutors, with good nutrition, with people who want to help them, or maybe in utero, the environment in utero is not nutritious, and so the person will exhibit a stunt in their intellectual capability. There are some diseases that are strongly affected by environmental and lifestyle. So some conditions are influenza and measles. Again, exposure to other people that have this condition will actually increase the likelihood of someone getting it. And then you've got multifactorial conditions like diabetes and heart disease that have got a genetic component. But again, it also depends on exposure to the environment, on lifestyle, on environmental diet, and exposure to smoke and other things to actually determine whether a person has an increased or decreased likelihood of getting diabetes or heart disease. And there's very few conditions, the conditions that we looked at, that are truly genetic, that are governed only by what you inherit from mom and dad. And these are referred to as single gene diseases, and examples of single gene diseases are cystic fibrosis, hemophilia, and as we talked about before, Huntington's disease, sickle cell disease. Those are single cell diseases that are single gene diseases that are strongly linked to the inheritance of genes from mom and dad, and not the environment or lifestyle at all. All right, I hope you've enjoyed this.